In 2002, a team of scientists decided to break the golden rule of ecology. The rule was simple. First, you clean the river. Then you bring back the wildlife. You do not put healthy animals into a chemical dump and expect them to live. But Dr. Anson Hines and his team had a different theory, the reverse order hypothesis. They argued that we did not need to clean the water to save the crabs. We needed to release the crabs to save the water. So they took thousands of hatchery-raised blue crabs and released them straight into the Lafayette River, a waterway so toxic that 90% of the fish had liver cancer. Critics called it ecological malpractice. They warned the crabs would be slaughtered by predators or poisoned by the sludge. They said it was a suicide mission. They were wrong. 5 years later, the suicide mission had not just survived. They were rewriting the rules of restoration. To understand the magnitude of this gamble, you have to understand the victim. The Atlantic blue crab isn't just a shellfish. It is the heartbeat of the Chesapeake Bay. It rules the food chain, cleans the river bottom, and drives a fishery worth hundreds of millions of dollars. By the year 2000, it was collapsing. Between 1990 and 2000, the population of the Atlantic collapsed. Adult female crabs declined by 84%, falling from nearly 80 million. Overfishing stripped the waters of breeding females before they could reproduce. Pollution choked the nurseries where their offspring should have lived. But the crisis was worst in the city rivers, specifically the Lafayette. This was not a habitat, it was a crime scene. For 3 centuries, it had been an industrial dump. The mud was laced with heavy metals and industrial toxins. The poison was so severe that researchers found liver cancer rates approaching 90% in the local fish populations. Runoff from the city fueled explosive algae blooms that sucked the oxygen out of the river bottom. In summer, dead zones formed where oxygen dropped too low for life. For a blue crab, which cannot swim away as fast as a fish, these zones were death traps. Crab jubilees, swarms crawling into the shallows just to breathe, became common. The underwater grass meadows had been wiped out by muddy water. Baby crabs had nowhere to hide. Without this grass, any baby crab that did enter the river was immediately eaten on the barren, muddy bottom. The river had become a killing field. Standard fixes had failed. Catch limits were not bringing the numbers back fast enough. Cleaning the water was taking too long. The problem was what scientists call a bottleneck. Even if there was food, the babies were dying before they could grow. Then, researchers proposed something radical. The proposal was simple, radical, and had failed before. If the river could no longer protect the crabs, humans would have to do it for them. The concept is called stock enhancement. It is not aquaculture. You are not raising animals to harvest them. It is biological reinforcement. Rear an army of predators in captivity. Deploy them to jumpstart a dying ecosystem. To pull this off, a massive coalition was formed, the Blue Crab Advanced Research Consortium. It sounded like a military operation. Led by the Smithsonian Environmental Research Center, the University of Maryland Center of Marine Biotechnology, and the Virginia Institute of Marine Science, the mission was to engineer a bypass for nature's broken cycle. But growing blue crabs is not like raising trout or salmon. You cannot just throw them in a tank and feed them pellets. Blue crabs are aggressive, territorial, and insatiable. In the wild, they are the hunters. In a confined tank, they are a cannibalistic nightmare. If you put 10 crabs in a bucket, you do not end up with 10 friends. You end up with one very large, very full crab. The first hurdle was getting them to exist. Researchers led by Dr. Jonathan Zohar had to close the life cycle in captivity, a feat that had stumped biologists for decades. They had to trick the crabs into believing it was spawning season by manipulating water temperature and light cycles, essentially hacking the animal's biological clock. They fed the microscopic larvae a precise and rich diet of rotifers and brine shrimp, babying them through their most vulnerable stages. The real mad science began in the nursery phase. To stop the crabs from tearing each other apart, the scientists had to design complex artificial habitats within the tanks. They used mesh screens and condos to mimic the missing seagrass, giving the juveniles just enough personal space to survive their own instincts. They were manufacturing a population that nature had stopped producing. The guiding philosophy was reverse order, but the tactical obstacle was recruitment limitation. The scientists believed that the river, despite its toxicity, could still support adult crabs. The problem was the babies. The larvae were too fragile to survive the journey from the ocean through the pollution. The river had become a fortress that no baby could breach. But the scientists knew something else. Blue crabs are engineers. They constantly dig and overturn the river bottom in search of food. The scientists believed that if they could release enough crabs, those crabs would dig through the river bottom, acting as a plow, aerating the sediment and clearing space for seagrass to return. They were betting that millions of years of evolutionary instinct could be replicated in a plastic tank. They were betting that the crabs would not just survive the toxic mud, they would fix it. The opposition to the plan was neither quiet nor minor. It did not come from a single loud voice, but from the rigid, established consensus of marine science. Three distinct groups stood in the way, each armed with data that said this project was doomed to fail. First were the institutional review boards. To them, stock enhancement was a dirty word. They pointed to the history books, specifically the failures in Japan and the US during the 1990s. For decades, governments had practiced spray and pray, dumping millions of microscopic larvae into the ocean and hoping for the best. The results were always the same: zero measurable effect. To these boards, this was just another expensive way to feed the fish. They argued you cannot solve an ecosystem collapse by simply throwing money at it. Then came the traditional ecologists. They argued that the researchers were effectively creating frankencrabs, animals that looked like blue crabs but acted like domestic pets. In the hatcheries, these animals lived in a utopia. They had no predators. They had constant food. They had never fought for territory. The ecologists predicted that these naive animals would be useless in the wild. They would not know how to forage. They would not know to bury themselves in the mud to hide. This fear was backed by the fisheries management councils. They focused on the cannibalism problem. They knew the blue crab is a cannibal. In the wild, adults eat juveniles. That is how the population regulates itself. Research indicated that in open water without seagrass, 75 to 97% of juvenile crabs are eaten. Releasing thousands of confused, soft, lab-raised crabs into a river full of hungry wild adults seemed like sending sheep into a wolf den. The managers argued they were not restoring the ecosystem. They were just delivering an expensive lunch to the existing predators. Then there was the location itself. Toxicologists called the Lafayette River a crime scene. The sediment was laced with industrial carcinogens known to cause liver cancer in 90% of local fish. Even if the crabs do not get eaten, would they slowly poison themselves? Would the heavy metals weaken them, leaving them vulnerable to diseases? This was a real and urgent worry. Finally, there was the fear of genetic pollution. Ecologists warned that if the hatchery crabs did survive, they could dilute the genetic diversity of the entire Chesapeake population. Or worse, they could introduce a disease from the hatchery, like the Rio-like virus, that could escape the tanks and wipe out the few wild crabs left. The risk felt existential to many. The consensus was clear. The review board said it was a waste of money. The ecologists said it was biologically impossible. The toxicologists said it was cruel. The smart move was to pull the plug. The scientists heard the warnings. They calculated the risks. They decided to do it anyway. The debate could have paralyzed the project for years, but the Chesapeake did not have years left. The fishery was in freefall. Time was running out. The decision to proceed did not come from one agency. It came from three institutions: the Smithsonian Environmental Research Center, the University of Maryland Center of Marine Biotechnology, and the Virginia Institute of Marine Science. This was not just a grant approval, it was a deployment order. But the approval came with conditions. biosecurity protocols so strict they seemed designed to make the project fail. The regulators knew the risks, so they built a cage around the experiment. First, they mandated a genetic lockdown. The biggest fear was that hatchery crabs would act like an invasive species, polluting the wild gene pool with weak DNA. To stop this, every single broodstock mother had to be wild-caught from the local Chesapeake population. The scientists could not import easier-to-raise crabs from North Carolina or the Gulf. They had to use the survivors of the crash to save the crash victims. Second, they established a disease firewall. In aquaculture, warm tanks are breeding grounds for pathogens. A single infected release could introduce a pathogen that would wipe out the remaining wild crabs. The protocols required screening every single batch for deadly pathogens. The rule was absolute. If one crab in a tank tested positive, the entire batch would be destroyed. The risk was brutal, but it was grounded in a new theory. The researchers argued that the spray and pray method of the past, dumping millions of microscopic larvae, had failed because the animals were simply too small to survive the ocean currents. Their solution was to grow them bigger first. They would grow the crabs past the bottleneck that was killing the wild population. It was a quality over quantity bet. Instead of releasing 10 million doomed babies, they would release 10,000 that could fend for themselves. The mission was clear: make this work without destroying what was left. They got the green light in late 2001. The team had their funding, they had their protocols, and they had their mandate. But they still had to answer the ultimate question: could a lab-raised crab actually survive in a toxic river? On a cold morning in 2002, the trucks were loaded. The debate was over. It was time to release the crabs. Between 2002 and 2006, the theory ended and the reality began. The release was not a single event. It was a sustained operation across the tributaries of the Chesapeake. The Lafayette and Elizabeth Rivers became testing grounds. The logistics were a nightmare. You cannot just dump crabs out of a bucket. Each release was 4,000 to 10,000 juveniles at a time. They were tiny, vulnerable creatures, transported from the pristine, filtered water of the hatcheries to the murky, industrialized river without dying of stress. But dropping them in was the easy part. Finding them again seemed impossible. The team knew that if they just released the crabs, they would never know if they survived. A dead crab leaves no skeleton. It just dissolves or gets eaten. To prove this gamble worked, they needed a way to track them inside the river. They turned to a technology that felt ripped from a spy novel: coded wire tags. Before leaving the hatchery, every single juvenile was injected with a microscopic magnetized wire in its leg muscle. These wires were invisible to the naked eye, but could be detected later with a magnetometer, a metal detector for crabs. Even if the crab molted and grew a new shell, the magnet stayed inside the muscle. For visual confirmation, they injected others with a colored polymer that glowed under the shell like a tattoo. They were not just releasing animals, they were releasing data. On the day of the first major release, the team, led by Dr. Anson Hines, motored out to the target zone. Some crabs were deployed onto barren mudflats where predators ruled. Others were released onto experimental oyster reefs restored by the Elizabeth River project. The scientists watched as thousands of their crabs drifted down into the dark water. The river was turbid, choked with silt and algae. It was effectively a blind drop. Once the crabs hit the water, they were gone. There were no cameras that could see through the muck. To get a glimpse of what was happening, Dr. Hines used ultrasonic telemetry. They attached tiny acoustic transmitters to the larger juvenile crabs and tracked them with hydrophones, listening for pings in the dark. The tension was immediate. The critics had predicted that the hatchery crabs would just sit there, confused, until a predator ate them, or that the toxic river water, laced with heavy metals and sewage, would shock their systems into failure. Now, the scientists could only wait. They had released 290,000 lives into a system that had spent a century killing everything in it. The experiment was live. The gamble was locked in. Now, nature would decide the winner. The first results did not trickle in. They flooded in. Within 48 hours of the 2002 release, researchers recaptured the first batch of naive crabs. Critics had predicted these animals would be starving, confused, or hiding in the wrong spots. Instead, stomach content analysis revealed something shocking. The lab-raised crabs were full, with clams, worms, and small fish. They were hunting just as well as wild crabs. The Franken-crab theory was dead on arrival. The real shock came when they crunched the long-term survival numbers. In the brutal mathematics of marine biology, the survival rate from larvae to adult is usually a fraction of 1%. Skeptics expected the hatchery crabs to perform even worse. They were wrong. By 2006, the hatchery crabs did not just match wild survival rates, they beat them. The data showed survival rates between 5% and 20%, numbers that would make any marine biologist do a double take. The second breakthrough came during the high-density releases from 2011 to 2013. This is where the gamble hit the jackpot. In targeted release zones, the local crab population did not just inch up, it surged by 40% to 60%. Then came the turning point. Scientists discovered why the crabs were surviving the toxic river. It turned out the toxicologists had missed something. The crabs did not care about the chemistry, they cared about the architecture. In areas where the Elizabeth River Project had restored oyster reefs, the crabs thrived. The complex shell structures protected them from predators, erasing the naive disadvantage. The experiment proved a new rule: structure trumps chemistry. And the reverse order theory. It worked. The crabs began foraging, aerating the soil, and clearing the bottom. Within a year, seagrass coverage in the river jumped from 15% to over 30%. The crabs were not just living in the river, they were fixing it. But restoring the habitat was only half the battle. The final test was migration. Survival means nothing if they do not reproduce. The team waited to see if the females would instinctively know to migrate south to the mouth of the bay to spawn. Months later, the reports came in. Watermen miles away were catching pregnant females bearing the telltale magnetic tags. They had navigated the industrial maze, survived the dead zones, and completed the journey. The scientific community, once loud with warnings, went quiet. The waste of money had just provided the blueprint for saving the species. The population in the Lafayette began to stabilize, not because nature healed itself, but because humans had successfully stepped in where nature could not. The critics had predicted a massacre. The data showed a resurrection. The gamble had paid off. The program wasn't cheap. It required years of high-tech hatchery operations, complex telemetry, and a logistical army. To the bean counters, it looked expensive. But the results tell a different story. The experiment did not just save crabs, it proved the river can be saved. The success gave the Elizabeth River Project the proof it needed to go further. By 2016, the river was removed from the state's bacterial impairment list. By 2018, it became the first tributary in Virginia to meet its oyster restoration targets. The crabs had proven the river was viable, and they gave the restoration movement credibility. Economically, the stakes were massive. This gamble helped stabilize a fishery worth hundreds of millions of dollars annually. But the most surprising profit was social. Watermen, historically skeptical of regulation, became active partners in the science. Over 4,000 residents joined the River Star Homes program to cut pollution at the source. The lesson was clear. Waiting for nature to fix itself does not work when humans broke it. They had to build the reefs. They had to grow the crabs. Was the mad science worth it? The Lafayette crabs proved something bigger. They proved that targeted intervention works. The question is not can we intervene, but are we willing to? The critics called it a waste of money. They said the crabs would die within days. They were wrong. The Lafayette River went from toxic wasteland to a thriving nursery, not by waiting for nature to heal, but by forcing it to. Sadly, blue crab numbers are down again. Still, the gamble did work. The blueprint still exists. The question is, will we use it again? If you think this experiment was reckless, or if you think it is exactly what we need more of, feel free to drop a comment. If you're interested in more videos about invasive species, subscribe because this is just the beginning.